Okay, so in this video we'll be discussing about the first of all we'll be discussing about the duodenum, that is the part of the small intestine. So this is the detailed lecture of the duodenum. Okay, so so duodenum is the first part of the small intestine which is short in length. It is about 30 centimeter long, and this is attached with the pyloric stomach, which means that, for example, this is a stomach. For example, this is a stomach, and at the end of the stomach, this is the pyloric sphincter is there. After the pyloric sphincter, there is a duodenum. This is a duodenum. Okay, so at the junction, here you can see that at the small intestine and duodenum, between that there is a sphincter that is called the pyloric sphincter, and duodenum is basically the C-shaped structure. Okay, so we were done with the introduction of the brief introduction of the duodenum too, but here we are discussing the detailed lecture with the layers, and you can say that the cells of the duodenum. So here you can see that we are having a, first of all, as you guys all know, we are having a 4 layers. First one is the mucosa. Okay, and this one is the submucosa. As you guys all know, the muscularis and then the serosa. These all are the same layers which were in the stomach, but their cells are different. In mucosa, the cells are different. Okay, but little bit there is a change. Okay, but their enzyme are different in the small intestine as compared to stomach. The pH of their you can see that enzyme are different. So let's talk about the mucosa, which is the first layer. Okay, so in mucosa you can see that they are the villi. You can see that these are the villi. Here the villi are present, brush border like structure. So there is a micro villi are present. Okay, villi means plural, and if you're talking about single, so there you can say that as a villus. So these are the micro villi. Okay, so what does it mean? It means they are the micro villi. They increase the surface area and for the absorption. Okay, so mucosa have 2 glands. Number 1 gland is the crypts of Liber Cohen. Okay, and in the mucosa we are having crypts of Liber Cohen. In the submucosa we are having a you can say that Brunner glands. Okay, so this Liber Cohen is present in all the small intestine, which help in the enzyme, and you can say that mucosa. Okay, so in submucosa their cells are present, which are these are the Liber Cohen gland in which the cells are present. We'll be discussing that too in the detail too. Okay, so here you can see that the brush border like structure is there, that is the micro villi, which help in the enzyme or mucus. Okay, so first of all, let's talk about the Liber Cohen, which is present between the two villi, and this is the glands of the mucosa layer. Okay, so microvilli are basically the, you can say that the finger like projection. Okay, now let's talk about the submucosa. So submucosa is having a, this is the submucosa. Okay, so submucosa is having a glands that are the Brunner glands. And Brunner glands, you can see this is this is the mucosa, this is the submucosa, and Brunner glands are present in bunch-like structure. Okay, bunch-like structure. Okay, and you can see that the mucosa and submucosa is having a same duct. Their ducts are same. Okay, but their duct will open into the mucosa. This their duct, some mucosa duct will open into the mucosa. Okay, and the secretion will mix up. Okay, so their both secretion is called the intestinal secretion, which is in glands they are cells, so their secretion are called the intestinal secretion, or you can say that as a succus extericus. Succus extericus, you can say that this is a secretion name, succus extericus. Okay, and if you're talking about the composition, so in composition there are enzyme, mucus, okay, hormones. Okay, so there are means you can say that in glands there are many cells. Okay, now if you're talking about the cells of crypts of Liber Cohen, so number one cells, they are the goblet cell. Goblet cell are also called the mucous cell. This was also in the, you can say that, stomach. So there in you can say that in the small intestine their pH is in the alkaline pH, which is 8. Okay, and you can say that if their pH is 8, so the chyme which is coming from the stomach, okay, so the chyme will get neutralized by this pH. Okay, so chyme will be neutralized by this pH. Number second cell is Pence cell. Pence cell basically secrete the enzyme, that secretes the enzyme. Enzyme, but which type of enzyme? Local enzyme. We'll be discussing that too, and you can say after that cells. Local enzyme we'll be discussing in detail. Okay. So intestinal enzyme, local enzyme, you can say. The third one cells are the enteroendocrine cell. Enteroendocrine cell that secretes the hormone, okay, histamine into the blood, secretes the hormone which is histamine, that into the blood, that is responsible for the vasodilator, and it work as a neurotransmitter. Okay, fourth one cell is the argentine cell. As you guys all know, argentine cell which secrete the serotonin, that was also in the, you can say that, in the stomach, that secretes the serotonin, okay, which was a neurotransmitter. Now the fifth one cells are the basal cells. Basal cells are basically the pre-existent cells, which mean new cells will generate by this. Okay, these all cells are the cells of Crips of Cohen. So these are the new cells. So they are you can say that the cells are similar to the stomach. So we have already learned in the detail. So these are the same cells. Only Pence cell is, you can say, the Pence cell and enteroendocrine cell. Enteroendocrine cell and Pence cell are different, but and all the cells are same like the stomach. So here the, if any cells is damaged, so new cells will be, you can say that, if goblet cell will be like will be produced by the basal cell. If Pence cell is damaged, so they will be produced by the basal cell. So they basically produce the new cells. So new cells will be generated by the basal cells. Now if you're talking about the duodenum, or you can say the mucosa layer, duodenum, okay, so duodenum in the mucosa, which is epithelial layer, okay, so it consists of two cells. Number one is exocrine cell, second is the endocrine. Okay, so exocrine cell basically secretes the enzyme. First of all, let's understand that what is exocrine. Exocrine means they are having a duct. They are having duct which is into the blood, but they are having no duct. they will be direct into the, they secretes their hormones into direct into the blood, but they'll be secrete anything enzyme, like they are having a secret enzyme, so the enzyme will be moving from the duct into the blood, but they are ductless, and they are having duct. Okay, they secrete enzyme. Okay, so like they secrete enzyme, they say they are having goblet cell, they are having a enzyme that is peptidase. Okay, which actually break the bond of amino acid. Okay, if you're talking about the local enzyme, that secret enzyme, local enzyme, I told you that we'll be discussing in the detail. So local enzyme are basically the enzyme which are, you can say that, the amylase, sucrase, okay, lactase, maltase, these are the enzyme. As you guys all know, in biomolecules chapter we have discussed it's all the enzyme in detail. Okay, maltase, lipase. Okay, so as you guys, you if you're talking about the you can say the amylase. So amylase is basically work on the starch, and starch break down into the maltose, as you guys all know. If you are eating a starch, so it through the starch will be break down into the maltose. If you're talking about sucrase, so sucrase is basically break down into the glucose and fructose. These are the sugar. Okay, now if you're talking about the lactase, so lactase is the, you can say that, the milk sugar. So this is basically break into the glucose and galactose. These are the sugar. Okay, lactase. Now if you're talking about the maltase, so maltase is the malt sugar which is break down into the two molecule of glucose. Now if you're talking about lipase is, these all are the enzymes. So lipase is the enzyme which basically which break the, you can say that, which is made up of the fatty acid and glycerol. Okay, in the biomolecules chapter we have discussed all these. So these are the local enzymes which are present in the layer of the mucosa. Now if you're talking about the endocrine, so endocrine cells basically secretes the hormones. These secretes the enzyme, these secretes the hormone. Okay, so hormones, so there are two hormones. There are two hormones. First one is the choleo cholecytokinin. Second one is the secretin. Okay, cholecytokinin are basically called the I cells, and this is called the S cells. These enzymes are having the work as G cell. As you guys all know, G cells was the gastrin cell. These work as the, you can say, the G cell or gastrin cells. So what was the work of the gastrin cell? That stimulate other, you can say that, the gland cells to start their secretion. So they work as the gastrin, and gastrin will work as to stimulate the local enzyme to produce their secretion. Okay, so they will work as a same G cells, cytokinin and secretin. They will work as same, and they will also stimulate the pancreas as well as gallbladder. Okay, so now if you're talking about the choleocytokinin, so they are, I cells are basically the intestinal peptone which release into blood, and from blood into the pancreas and bladder, which is gallbladder. So the peptide hormone is there which work on the protein. So basically they contract the, this enzyme, this hormone contract the gallbladder to produce their secretion. Now if you're talking about the you can secretin, so secretin are called the S cells. So they basically release into the blood, and they are having no duct, because as you guys all know, they are the endocrine hormone. So they basically release in the, they also release into the pancreas and gallbladder. Okay, so they basically activate non-enzymatic part of the pancreatic juice, which release, like for example, the ions, that is sodium, potassium. Okay, so these basically secrete the ions to reduce the acidity. You can say that acidity kind, acid kind, to reduce the acid kind. Okay, so they are having a, you can say that they are having a pH alkaline. So if when chyme is there, when chyme will be coming from the stomach, so they will basically neutralize the chyme, and the pH will of the chyme will convert it into the alkaline level. So as you guys all know that, so these both enzyme will stimulate when acid chyme will be coming. Okay, when acid chyme will be coming, so it will become, so their secretion will be coming like from the gallbladder and pancreas. The secretion will be coming from the hepatic duct into the duodenum. So both cell will become down at the pH of 4 pH. Okay, and they release their secretion, and the secretion will, you can say that their secretion will neutralize the pH of the chyme, and then the chyme will convert into the pH that is alkaline. Okay, so in the duodenum there these two cells basically work. Now if you're talking about the, in the exocrine cell, so in the exocrine cell there is a enzyme that is the enterokinase, which is also called the enteropeptidase. Okay, this is present in the cells, that is crisps of Cohen. Okay, okay, so they are around about 7 to 8 pH. This is basically the enzyme which will work in the mucosa layer. So they will convert the trypsinogen into the trypsin. Okay, so as you guys all know, in your stomach, HCl was working to activate the inactive enzyme. So here the enterokinase will be work as a HCl. Okay, HCl which will be like which will active the inactive enzyme. Okay, so enteropeptidase, or you can say that this is the enzyme. So trypsinogen is basically coming from the pancreatic juice. So enterokinase is an enzyme which, this is basically inactive, so it will convert into the active trypsin. Okay, so this trypsin will work in the protein, in the digestion of the protein. Okay, so these were the enzyme which work as a, you can say that, and lipase was the enzyme which was work as a, you can say that to break down the fatty acid and glycerol. As you guys all know, we have discussed this here. So today we have discussed about these all. Okay, now in the next video we'll be discussing about the liver, the detailed lecture of the liver.